Hello everyone, today I'm going to be presenting CONCERTO, a graph neural network approach to molecular carcinogenicity prediction. My name is Philip Fradkin, and this work was done with my collaborators Adamo Young and Lazar Atanaskevich, and supervised by doctors Brandon Fry, Leo J. Lee, and Bo Wang. So we can think about carcinogenic compound discovery in two main modes, the high effect size mode and the high exposure. So on the left you see a picture of a plant that contains aristolochic acid that proved to be one of the most carcinogenic compounds tested. And on the right you see a picture of an English chimney sweeper that in 1775 Percival Pott identified to have high rates of squamous cell carcinoma as a result of their exposure to soot and ash. So one of our motivations for this project of predicting compound carcinogenicity was to detect molecules that are moderately carcinogenic and have lower exposure but are still harmful to people. So our contributions are we reinterpret carcinogenicity prediction as a regression problem instead of a classification problem, and while taking into account compound dosage. We're the first to apply graph neural nets to carcinogenicity prediction. We pre-train on related but lower quality mutagenicity experiments, and we conduct transfer learning from a large self-supervised model. And finally, we generate counterfactual molecules for model interpretation. So there are two main methods of measuring molecular carcinogenicity. The first is through carcinogenic experiments, which are typically conducted in rodents, and although they are pretty accurate and informative, they're quite expensive and slow, so there aren't that many samples. And on the right, you see mutagenicity experiments, which are conducted in bacteria, and they only measure genotoxicity effects and have lower rates of replication, but they're cheap and abundant. So here you see a representation of our model, which consists of three main parts. In blue, you see this Grover GNN transformer part, which was released in 2020, and it's a 100 million parameter model trained in a self-supervised manner. In green, you see the second part, where we take a molecule fingerprint representation and train a multilayer perceptron on the fingerprint representation as well as the Grover representation. And finally, our last part, we pre-train using mutagenicity training and fine-tune on carcinogenicity data, and do this multiple times, and we find that this improves performance. So in our evaluation, we first test our model performance on the CPDB test set, and we find that CONCERTO outperforms two of our baselines, which is part one, which is the GNN transformer baseline, and part two, which is the fingerprint baseline. And CONCERTO outperforms both of them, and we find that on CCRIS, an external test set, CONCERTO performs better than our baseline. In addition, we evaluate against Carcinopred EL, which was also trained on CPDB, which is an ensemble-based fingerprint method. Finally, to interpret our model predictions, we utilize counterfactuals, and these are samples that are close in the input space but far away in the prediction space to a sample of interest, and we identify those using XMOL. And we consider Tanimoto scores as a measure of molecular distances. So in this plot, you're able to see on the x-axis the molecular distance to the molecule of interest, and every single other point is a perturbed molecule. And on the y-axis, you have the carcinogenicity percentile. So we're interested in identifying these counterfactual molecules that are related to our original molecule, but the model interprets as either extremely non-carcinogenic, like this point in blue, and extremely carcinogenic, like this point in red. So we hope that we think that it's important to think about compound dosage while assessing model performance, while assessing carcinogenicity performance, and we hope that this counterfactual method can help people interpret our model predictions. So, and in data constrained settings more broadly, we think that large models trained with a self-supervised learning objective, pre-training on related experiments, and encoding domain expert knowledge are all helpful. Thank you, and if you're interested, please check out our preprint.